Welcome to solverfy.
Hello students, the question says that the stickleback population recently became isolated in freshwater lake from its marine ancestral population, and the phenotype of the salt tolerance being strongly selected against in the freshwater population. The salt tolerance phenotype in this typholite species is under the control of a single genetic locus, which is denoted as S, which has two alleles. One is S1, which is the dominant allele. Another is S2, which is the recessive allele. And the dominant allele does not tolerate salt, while the recessive allele does tolerate salt. And the homozygous recessive genotype, which is S2 has a fitness of 0.001 relative to that of the other phenotype, other genotype, that is the selection coefficient of 0.999 on S2 allele. Now the question also says that if the initial allele frequency of S2 is 0.85 in marine ancestral population, what is the allele frequency of this allele, which is S2, after two generations in freshwater population? First, we need to find the initial genotype frequencies of the in the freshwater population, and we can use the Hardy-Weinberg equations, Hardy-Weinberg equilibrium to do this. The formula for the Hardy-Weinberg equilibrium is p square plus 2pq plus q square equals 1, where the p is the frequency of the dominant allele and q is the frequency of the recessive allele. So we are given that the initial frequency of S2, which is denoted as Q, because it is the recessive, as 0.85. So this is the Q, as we know that P plus Q equals 1. So we can calculate the P, which is the allele frequency of S1, as 1 minus 0.85. So P is 0.15. So this is the allele frequency of the dominant allele, and this is the allele frequency which is given of the recessive allele. Now we will calculate the genotype frequencies of S1, which is the dominant gene, is represented as P square. So P is equal to 0.15, so we square it, which will give us 0.225, the genotype frequency of dominant gene, while the genotype frequency of the recessive gene is 0.85 square, which is 0.7225, and the genotype frequency of the heterozygous, which is represented as 2pq, is 2 times 0.15 times 0.85, which is 0.225. So now let's find out the fitness of each genotype, which is the S2 genotype, which is already given as the fitness is 0.001 relative to other genotypes, since the other genotypes have a relative fitness of 1, relative fitness 1. So we can assign the following fitness values, which is for dominant homozygous, it is 1. It is 1, and heterozygous, which is S1, S2, also 1, and S2, S2 is 0.001. Now we can calculate the expected genotypes or genotype frequency after one generation of selection. So the expected genotype frequency for homozygous dominant will be the genotype frequency times the fitness, which is 1, upon the fitness of all the genotypes, which is given by the respective genotype frequency and the fitness. So it comes out to be for homozygous dominant gene as 0.0806. Likewise, the expected genotype frequencies for homozygous recessive is 0.0026 and heterozygous S1 S2 is 0.9148. Now we can calculate the new allele frequencies after one generation, which is for S1 it will be 2 times 0.0806, which is here for S1 S2, plus the frequency of heterozygote, which is 0.9148 upon 2, which comes out to be 0.053. This will be the new allele frequency of S1 allele, and for S2 allele likewise 2 times 0.0026, which is 0.0026 plus 0.9148 from heterozygote upon 2, which comes out to be 0.462. So we need to repeat this step 3 to 6 for second generation, and first find the genotype frequencies using the new allele frequencies which we calculated here, and then using the fitness we can calculate the new allele frequencies for the second generation. I hope you found the video useful. Thank you.